Well, good afternoon. Welcome back to Turk's Science Speaks Fall Seminars. My name is Jeff Schlatter, and I serve as the director of the UC Davis Tahoe Environmental Research Center, or TURC. If you want to learn more about TURC, the research we conduct here and around the world, the public education we provide for the local and regional community, and our work with agencies here in the Tahoe Basin, please visit our website, tahoe.ucdavis.edu. You can also find out how to follow us on social media and how you can join in with and support the work we do. One of our goals is to facilitate interaction, dialogue, and discussion. Easy to say, but challenging to accomplish when we need to stay distant from each other and rely instead on virtual platforms to bring us together. We will be attempting to address your questions at the conclusion of today's presentation via the Q&A function on Zoom. Please don't yell out questions because we won't hear them. Also, don't use the chat function to add questions because we won't be monitoring that. Instead, type your questions into Q&A, which is at the bottom of your screen, and we'll try to get to as many as we can. We are fortunate today to have two excellent scientists for you. Before I introduce them, I wanted to say that our next Fall Science Speak seminar will be in exactly 4 weeks on December 10th and is titled Local Renewable Energy Transition Planning and Climate Resilience. Okay, it's now my pleasure to introduce Jenessa Gjeltema and Katie Senft. Dr. Gjeltema is an assistant professor in the School of Veterinary Medicine at UC Davis and scientific research advisor to the Sacramento Zoo. She earned her Doctor of Veterinary Medicine degree from North Carolina State University. Among her many interests are the effects of environmental plastic contamination on animal, human, and ecosystem health. Katie Senft graduated from the University of Nevada, Reno in 2007, was an AmeriCorps member with TURC in 2009 before joining our field research team the following year. Since then, she has spent most of her career either on or in Lake Tahoe, collecting and analyzing samples for lake health, invasive species, and most recently, microplastics. As you all know, the prevalence of single-use plastics around the world has grown tremendously, and once their use has been fulfilled, many of them naturally move downhill and end up in the low points in the landscape. That eventually becomes our oceans and our lakes. With an annual visitor population in the many millions, we should all be wondering about the plastics in Tahoe and where they might end up and what that might mean for us. So today's presentation addresses precisely those topics. It's titled Studying the Fate of Microplastics at Lake Tahoe, and I'll now transfer control of your screens to Dr. Gjeltema.
Thank you so much for that wonderful introduction. Very excited to be here today to chat with you guys about one of my most favorite topics to discuss. Let me see if I can get my presentation going here. Okay, hopefully you guys can see that. All right. So I'm going to be giving you a little bit of an introduction to microplastics, kind of talking a little bit about what they are, where they come from, and why we might be interested in studying them. I'll talk a little bit about how we actually study them in our lab, and then we're going to switch tracks, and Katie Senft is going to talk to you a little bit about what we're doing to evaluate microplastics at Lake Tahoe specifically. So you're all here because you want to learn a little bit more about microplastics, specifically at Lake Tahoe. And I'm going to, discuss some of that, shed some light into what microplastics are, where they come from. But I think before delving into the topic of microplastics, it's important to take a step back and talk about plastic. So the term plastic essentially means being capable of being shaped or molded. And it's colloquially used to refer to synthetic man-made polymers that have the property of being able to be shaped into a variety of different forms. But what actually is it? So plastics are generally composed of synthetic polymers, and you may have heard them be referred to as polymers in the past. Um, and they're basically composed of repeating subunits, individual units, which is represented here by a blue sphere. Those subunits are joined together to form polymers, which you kind of see in this representation below the monomer. So you basically have the same repeated unit bound together. And these make up an individual molecule that's relatively large. They're kind of these long chains with a carbon backbone. And when they come together with a variety of different additives, they have the properties of what we, colloquially know as plastics. Depending on what the subunit is that's selected, so this subunit here will determine a lot of the properties of a plastic. It can determine how rigid it is, how durable, its color, its flexibility. So that in combination with the additives that are mixed into the polymer can alter its characteristics to make them useful in a variety of different ways. The vast majority of our basic polymers are made from petrochemicals, which is oils, and are that is the source of most of our basic plastics that we currently use. Although other derivatives or sources of plastics are definitely being developed and are possible. So the different subunits of microplastics and these different polymers correspond to its name and what many of us are more familiar with, which is the resin recycling codes that are sometimes found on the surface of a plastic material. So most synthetic polymer plastics are initially made into pre-production pellets, which you can see here in my hand. This is just a handful of small pellets. And these are sold to manufacturers, and those manufacturers will then shape and mold them into the various products or forms that we eventually see them in. They can be made into films, they can be extruded into fibers or filaments. They can also be made into, woven into clothes and textiles or used to coat surfaces. So they're used in a variety of ways, and depending on what the characteristics are needed of that plastic, the different subunits are selected for that. It's important to note that not every plastic is thermodynamic or thermomoldable. And that's important to note that, not all of these plastics are recyclable and not all of them can be reused. So what do plastics turn into? And I think most of us are familiar with some basic ones, but there might be some uses that you may be less familiar with or not really realize, not really think about it as being a plastic. So most synthetic clothes are made out of plastic. Textiles and toys, plush animals are often made out of plastic. Cosmetics can contain plastics, not just in their packaging, but actually in the cosmetic itself. A lot of our personal hygiene items, diapers, baby wipes, masks, disposable masks, and healthcare tools, including syringes and needles, IV lines, and a lot of the tools that are necessary to provide healthcare are currently made out of plastics as well. One-time use and convenience goods, as well as food wrappers, are also used or produced out of plastic. And most of us are familiar with some of these packaging items, bags, and wrappers. They're also used for a variety of industrial processes, for abrading surfaces. So they'll kind of have small microbeads that they'll spray onto surfaces to remove external paint or to kind of sand. They can be used for tires and various automobile parts, as well as for construction. So vinyl siding, piping, and flooring are all uses that we are have around us every day, but we may not necessarily put that together with the idea of a plastic. So clearly, plastics are used for a variety of different purposes. And I think, most of us don't love plastic. However, I would say nearly all of us love what plastic can easily provide, whether it's convenience, hygiene, protection, comfort, fashion, friendship, being with family. All of those things are usually things that most people value. But ultimately, the usefulness of most of our plastic products, that value ends, and the usefulness of those plastic products end. The package is opened, the sandwich is consumed, the tire is worn down, and the home is renovated. And so what happens next in that life cycle? Well, for some plastics, they're able to be recycled. But as we discussed, there's a large number of plastics that are not able to be recycled, and that end up disposed in various ways. So that might be packaging up that plastic in more plastic and disposing of it through municipal, landfill or waste disposal methods. It can also be lost into the environment. So some of this is just almost imperceivable, as in with construction, when you're cutting into a piece of plastic and you've got particles that are, coming off of your work, you may not even realize that they get released into the environment. Sometimes they're intentionally disposed of into the environment inappropriately. Sometimes they're just blown away and end up kind of entering either our air, our land, or our waterways, unintentionally, just as a side effect of them being used and accidentally disposed of in an inappropriate way. So that takes us back to microplastics. And this is really where microplastics come from. So although the chemical decomposition of plastics, it's a very, very slow process. And so plastics in general tend to break into smaller physical pieces long before they break down chemically. So the chemical breakdown of plastics can take anywhere from just a few years to up to millions of years for that actual chemical structure to break down. In the meantime, different factors can actually enhance that larger piece of plastic to break into smaller and smaller pieces over time. So things like UV light exposure or physical contact or movement, even temperature changes can contribute to sort of wear and tear on plastics that are in the environment, causing them to become microplastics. So there's so many different kinds of microplastics out there. In general, when we're talking about a microplastic, we're talking about something that is less than 5 millimeters in size, but that they go all the way down to about 1 millimeter in size. And they can get even smaller than that, but usually we refer to those as nanoplastics, which is just a smaller size fraction. They can come in a variety of different shapes. So, a lot of people think of microplastics being microbeads or microspheres that had received some publicity in the past for being in, hand washes or cosmetics. But there's a lot of other kinds. Fibers are becoming more prevalent and more interesting from a biologic perspective. We also get fragments and films. So there's a lot of different shapes that microplastics can end up in. And there's, an ever long list of different types of microplastics as far as their chemical composition and what that subunit really is structured by. So they also can come in a variety of different colors, which may attract certain animals to them, or those dyes inherent to that color may have certain effects. So there's a lot of different combinations, and no real two microplastic fragments or particles are the same. And we don't know yet which of these, sizes and shapes and types are going to be the most, have the most potential risk to cause effects in from a health perspective. So let's talk just a minute about the health, potential health risks for animals, humans, and ecosystems, because that's what, we're very interested in as far as figuring out, what are the concerns, and one of the reasons why we need to evaluate how much of these microplastics are in the environment, and pairing that with what the risk is for, the health of animals in our, habitats, as well as human health and ecosystems. So there are several different categories of health risks. Some of those can be physical effects. So the plastic particles might accumulate on the surface of certain types of animals and impair them from functioning normally. They can also be eaten and cause irritation to the gastrointestinal tract, or potentially obstruct the gastrointestinal tract, blocking it up. They can also potentially spread persistent organic pollutants and heavy metals. So oftentimes they can act like a sponge, and pollutants can be sort of attached to the surface of microplastics, which can sometimes enhance, their ability to be exposed to a certain animal who might be predisposed to eating that type of plastic or that size or shape of plastic. And so it can interact with other contaminants of concern in the environment. It can also potentially act as a vector for pathogens. So as we learn more, we find out that there are colonies of bacteria that will aggregate on the surface of a lot of these microplastic particles. And they can potentially harbor harmful bacteria, as well as parasites, or as we discussed, can be vectors for the pollutants. So they can serve as sort of a vessel for delivery of different harmful materials or microorganisms, which is a potential health risk that we should be paying attention to. They can also leach known carcinogens or other toxins. So as we talked about, additives are oftentimes incorporated into the polymer itself, the synthetic polymer, in order to change its characteristics. And some of those additives are directly toxic. And so as they end up releasing from that microplastic particle that can lead to exposures of animals that might be in contact with that particular piece of microplastic. So now that we've talked about why we might want to pay attention and study microplastics, I just want to share a little bit about how we go about studying microplastics. So step number one is generally a collection of samples after a well-designed research study has been proposed. The samples can be a variety of different things. They can be environmental samples. They can also be biologic samples. So we might be sampling for ingesta or feces, tissues. And so those samples can be composed of a variety of different substrates or primary components. And then the actual microplastics in those can definitely differ in size as well as type. And so depending on how large of a microplastic we're looking for, we might have different ways of isolating the suspect particles, which I'm going to talk about in just a second. Once we're done isolating those particles that we're interested in, then we can use a variety of different techniques to characterize and identify the chemical composition of that particle. Once we have all of that information, we can use that to then analyze whether there are relationships or where those microplastics are found the most to help us understand and be able to draw conclusions about what that means for animals, humans, and ecosystems. So on the right side of this picture is actually a microplastic sample. This is a sample that was obtained from a trawl. So a net was pulled through the water, and someone handpicked each one of these potential microplastic particles. And we separately identified and characterized each of these particles. And so this was an example of a hand-selected and isolating procedure. But there are also the more complex samples that might not have plastics that are large enough to see. And in those cases, we might have to process that sample. So for those samples, we might have to digest the organic material from the sample first to remove any of that organic material that might be impinging on our ability to really get to those microplastics and analyze them. We then can do separation using different densities to obtain a fraction of the sample that just contains the microplastics. And what that allows us to do is to separate those. We can either put them onto a filter or a petri dish and then analyze those with our instrumentation in the lab. So in our laboratory, we actually use Raman microscopy, microspectroscopy, where it actually utilizes some aspects of chemistry. So a lot of the chemical bonds that exist will cause a vibrational movement in a molecule. And we can actually use that because when you shine a laser beam on the surface of that chemical, it can cause a change in the wavelength of light or the color of light that is reflected off of the surface of that particle. So for us, we'll actually measure what kinds of light are reflected out off of the surface of a particle of interest and measure that in order to, get this representation of what the chemical structure of that particle is that we're interested in identifying. So to do that, we are basically measuring the intensity of different wavelengths of light. These wavelengths correspond to different colors. So essentially, we are measuring rainbows that reflect off the surface of the particle. We make a graphical representation of that. So here we've got light intensity and wavelength. So we get this nice signature of what that underlying chemical structure is. We can then compare that to a library we have of known chemical signatures to help us identify what that underlying chemical structure is. And so that allows us to determine whether it's, one plastic versus another type of plastic, or not plastic at all, which can also happen. So this is a graphical representation of what this process actually looks like when we graph. So you can see this black line represents the chemical signature that we're measuring of a type of microplastic that we found in one of our samples. The red line actually represents our library, and we're basically comparing those two to see where they match up and see how close of a match our particle is compared to what we know from materials that have already been analyzed in the lab. So this is what the process actually looks like when you're doing it. So this image up here at the top is actually a bunch of microplastic particles that have been filtered out onto a specialized filter and put onto a microscope slide. We can then put that slide onto our microscope. We usually mount the filter to kind of smooth it out, makes it easier for us to see. And then we can take a look at the filter surface. So this is actually a picture of the filter surface that we're looking at here. And as you can see, there's a lot of different small particles on the surface of this filter that are very tiny that we can then analyze individually to determine what their composition is. We're also able to use software to our advantage to help us speed up the process. So we can actually locate and identify using some automated processes. So these are actually really tiny, probably the smallest size that we can analyze on our system, about 1 micrometer in size particles that are microplastics. And I'm just going to blow this up for you so you can kind of see what it looks like. So here's your particles of microplastic on the surface. You can actually see some of the holes in the filter kind of peeking out, but those are just holes that allow the water to drain through when we're processing that sample. So this is actually the software that allows us to speed up the process by automatically detecting plastics or particles of interest in our sample on the filter. So here you can see that each one of our particles is numbered separately to allow us to analyze each one as an individual particle and determine its characteristics. So what kinds of information can we get from analyzing these chemicals in the lab? We can determine their chemical composition. We can also determine the type of polymer or plastic that the particle is. We're also able to determine how much of a plastic might be in that sample. So we can determine the mass by weighing the sample. We can also determine the number of particles that were ultimately filtered out from that sample. We also can determine the shape and size because those may have implications for different kinds of health problems. Smaller particles may have greater surface area, which might make them more relevant to transporting different surface contaminants that might be stuck to the surface, or different shapes might be more prone to causing local irritation or physical disruption. So that kind of information is helpful for us to determining what types of risk might be associated with a certain collection of microplastics. I also think it's important to highlight how important it is to maintain quality in research, especially as it pertains to microplastics. So, for our research, we use a variety of tools to help us make sure that our quality remains high. That includes avoidance of use of plastic tools and PPE during our processing of our samples. We also use environmental chambers to prevent dust from accumulating or preventing our samples from blowing away. We have fastidious lab hygiene. We also use ongoing method validation, and we also are very transparent in our reporting. So that is all as far as our brief introduction into what microplastics are, why we might be concerned about them, and then how we analyze microplastics in the lab. I really appreciate your attention, and we're going to switch gears now. Katie Senft is going to talk to you about our specific research at Lake Tahoe, and we'll be happy to answer any questions at the end of the presentation.
Thank you for that great intro, Jenessa, to plastics and microplastics and how our samples will be handled once we get them out of the field and back to the lab. So let's see if I can get my presentation up and running. All right, I'm hoping everyone can see that. So I will be talking about our microplastics at Lake Tahoe. So Lake Tahoe is unique beyond its blue clear waters. It's also unique because starting in 1968, all the wastewater has been transported out of the basin. That was originally started because we didn't want a lot of nitrogen and phosphorus being loaded into the lake, which can cause algae blooms and loss of clarity. But in hindsight, it actually also prevented the release of microfibers into the lake that are produced when we wash our clothes and other, things in our house. So it was a twofold benefit when that happened. So we first started our work on Lake Tahoe microplastics in 2018, looking at plastics at the beaches. So we were curious to see if microplastics were at detectable levels in the lake. We have a great summer internship program, and so we thought this would be a really good project for them to work on. We decided to look at beaches because they're frequented by both locals and visitors, and we really wanted the public to be able to connect with our work. If we were sampling out in the middle of the lake, some plastics out there, it matters, but not as much maybe as if you're looking, picking around in the sand. We're at the beach and you can actually see these plastics and you can touch them and see that there are problems with your own eyes. And also ease of access. Beaches are easy to get to for us, and since our first project was unfunded, we wanted something that we could easily get to. So for this first study, we looked at beach sediment from four beaches around the lake. So we looked at Hidden Beach on the North Shore, Commons Beach, which is in Tahoe City, Lester Beach, which is located in D.L. Bliss State Park, and then Baldwin Beach on the South Shore. At each of these beaches, we laid out a 100-meter long transect line along the water's edge, and we put down a 1/4-meter quadrat, and we removed all of the sediment from the top 5 centimeters and put it in a glass jar and took it back to the lab for analysis. So you can see our wonderful summer interns here, Hannah and Maddie, working very hard at the beach, collecting the sand to bring back. Once we had our samples in the lab, we used that density separation and chemical digestion that Jenessa was talking about to isolate microplastics or microparticles from the beach sand. So this is a graph of what we found. So we collected a total of 5 samples from each beach, but just due to time constraints, we were only able to process 3 from each site. So the big takeaway here is we found plastics at every beach that we sampled. Not necessarily every sample that we collected had plastics in it, but the majority of them did. So this was a one-time sampling event that we did in 2018, and during that one-time outing, we found that Baldwin Beach had the most plastics, so about 120 milligrams per cubic meter of beach sand. I was surprised that Hidden Beach actually had so few that we found, because in the wintertime, when I've gone down there, there tends to be a lot of plastic and trash washed ashore. So since this work was done in the summertime, it would be interesting to repeat this in the wintertime and see if there's some seasonal differences between summer and winter at these different beaches. So now that we know that there's plastics in Tahoe, we want to find out where they're going. We know they're at the beach, but this brings us to our current work. So our 2020-2021 Lake Tahoe plastic sink study. So we're going to be looking at 5 potential sinks for plastics in the lake. So we'll be looking at the water column in municipal waters, deep water sediments. We're also going to look at a couple animals to see if they're ingesting plastics. And we're going to look at the surface waters as well. So for our water column sampling, we're going to be using what's called a Van Doren sampler to collect water. So you can see a photo of that here. It's basically a piece of pipe with plungers on either side. You can set a mechanism so it stays open. You drop it down to your desired depth. You send a weight down the cable, it closes it, and then you have a nice discrete sample of water that you can bring up to look at. So we're going to look at 6 different depths from the surface all the way down to the bottom of the lake. And the reason why we want to do this is different plastics have different densities. So some are light and they will float on the top of the water, and heavier ones can settle out, or maybe if they get a biofilm on them, that could cause their density to change and for them to sink as well. So we want to get a good idea of plastics that are distributed throughout the water column. So we'll be doing Raman analysis on these particles that are larger than 1 micron, and we'll be doing this quarterly in the lake. We'll also be looking at municipal water samples. So we'll be collecting water on the North Shore from the Incline Village Water Treatment Plant and from the Edgewood Water Treatment Plant in the South Shore. And this is with support from the Tahoe Water Suppliers Association. And we will be looking at, once again, particles that are larger than 1 micron, and we will be doing this sampling 4 times over the next year. We also want to look at deep water sediments in the lake. So we're going to use this box core sampler on the right to capture a big column of sediment from the bottom of the lake. We're actually able to get out on the water and do this in September, and you can see our nice mucky samples from deep in the lake. So these will also be analyzed for particles larger than 1 micron. And the deep water sediments are of interest because our lake is so deep. If we have these plastics that settle to the bottom, it might be kind of a way for them to be locked away from human consumption or wildlife. So it might kind of be more of a permanent sink for plastics in Tahoe. We're also going to look at animals in the lake. So filter feeders are at a greater risk for ingesting microplastics, and that's because they're non-selective feeders. They take in water, but they're not necessarily, picking out zooplankton or algae. They're just taking it all in and keeping what they believe to be food. So in China, they found that Asian clams are a good bioindicator of microplastic pollution in the local sediments. So we thought it'd be interesting to look at the Asian clams in Tahoe to see if that's true here as well. And we're also going to look at kokanee salmon. So they feed on zooplankton in the lake. We also consume them. And then other top tier predators in the basin like bears, eagles, as well. So we want to know if microplastics in the lake could be moving into the terrestrial food web. And finally, we'll be looking at surface water sampling. So this is a picture of a manta trawl, and this is a common sampling technique in marine environments. So when you hear about like the Great Pacific Garbage Patch and people sampling out there with nets, this is the kind of net that they are using out there. So in Lake Tahoe, we're going to be doing our surface trawls off the Tahoe City shelf. So we're going to be sampling at 2 depths, one at the surface, and then another one at 20 meters deep through the zooplankton layer. So once again, we want to look at those 2 different depths because different plastics, different densities. We want to know what's in the water column. And then it's also interesting to see what kind of plastics are in that zooplankton layer to see if that matches up with any plastics we might find in the kokanee, because that's where they're feeding as well. And each of these transects will be about 3 kilometers in length. So you can see a picture here of a couple samples that we collected in February. So this is our surface sample, looks pretty clean to the naked eye. And then this is our sample through the zooplankton layer. And you can see that it is just bright red with diaptomous zooplankton that are really common in the lake in the winter and the springtime. Once we get our net back on board, we use a deck wash to rinse all the everything that we caught to the bottom into the caught end of the net. Then we pour it in a sample jar and we take it back to the lab for processing. So here in-house for our surface water sampling, we do chemical digestion and the density separation, and we separate out all those microparticles, which are mounted on a Petri dish and sent down to Jenessa's lab for validation with the Raman Microspectroscopy. We will be doing this monthly for the next year. I believe our first sample was collected in August, so we will continue through July of 2021. But we did do a practice run in February to make sure we had everything down as far as field work goes in the lab. So we got some preliminary results that I wanted to share with everyone. So a couple words of, I guess, caution is these are preliminary results and it is from one sampling event. The data that I show you will kind of change after we have 12 sampling events to look at, but it's always fun to share some data that's hot off the press with everybody. So for our February sample, you can see here in the Petri dish, we found 60 microparticles in it. And so if you do the math, that calculates out to about 28,000 microparticles per square kilometer of lake surface area. Now that might sound like a lot, but when you compare it to other lakes or marine systems, it's not too bad. So I realized that Lake Erie and the San Francisco Bay are quite different. Obviously, they have larger watersheds, they have a lot more people, they have different industry, but there hasn't been a lot of work done on microplastics in freshwater systems. So it's hard to find a lake that's comparable to Tahoe that's had microplastic research done to kind of compare. So these were the ones I found. For Lake Erie, they did 8 different samplings and they found about 105,000 microparticles per square kilometer of lake surface area. And then in the San Francisco Bay, with 32 sampling events, they found about 270,000 microparticles. So you can see here a comparison of our Petri dish of samples and then a Petri dish from what was collected in the San Francisco Bay. And you can see that they definitely had a lot more particles to analyze than we found in Tahoe. So I also did a little bit with the particle shape. So Jenessa talked about the different shapes, films, fragments, fibers, foams, and spheres. And comparing Lake Tahoe to the Bay, I thought this was really interesting. In both systems, films, foams, and spheres, not very common. The most common shapes were fragments and fibers. And in Lake Tahoe, the most common shape was a fragment. whereas in the bay, it was a fiber. And I think this might be attributed to the fact that in Lake Tahoe, since our wastewater is transported out of the basin, we're not getting all that microfiber loading that other systems are getting. Whereas in the bay, all their wastewater goes into the bay, so they have a lot more fibers present. So it'll be interesting to see how this graph might change once we have more sampling events, but just really interesting stuff right out the gate. Jenessa was also able to do some Raman analysis to look at the polymer type of the samples that we got out of Tahoe. So the most common polymer was polyethylene. And then we also had some polypropylene, polyesterine, some nylon, and ABS, which I cannot pronounce off the top of my head. So we're just going to keep it as that. But there's 5 different types of polymers that were identified in the Lake Tahoe samples. And then we had about half the particles were of unknown composition. And with the polyethylene, it's the most widely used plastic in the world. So it's not surprising that it was also the most common type of plastic that we found in our Lake Tahoe samples. So we're going to monitor these 5 different potential sinks of plastic in Tahoe over the next year. And we hope to have a final report released late in 2021. So hopefully we will get to do another lecture shortly thereafter and share the results of our study. In the meantime, the future of Lake Tahoe microplastics. I found this little cartoon when I was on the web yesterday, and I don't think Tahoe is heading in this direction at all, but there's a lot that everyone can do out there to make sure that this cartoon does not come true. So what can you do? Probably the best thing you can do is reduce your plastics consumption. So reusable products like water bottles, bamboo cutlery, reusable Ziploc bags are all a really great place to start, or refusing single-use items like straws. So if we aren't consuming and using those plastics in the first place, they can't get into the environment. Recycle. Only 9% of the world's plastic is recycled. So try to make sure your plastic gets in the recycling bin. So if it is recyclable, hopefully it can get reused. Participate in or organize your own litter cleanups. So it's easy to go out and pick up trash on your own. Once the pandemic is over and we don't have to do social distancing anymore, there's a lot of great organizations locally to get involved with. So the League to Save Lake Tahoe has the Tahoe Blue Crew, and they organize beach cleanups. They do a big one every year on July 5th. Also Clean Up the Lake. They're doing an underwater cleanup of Lake Tahoe next summer. But this past fall, they did a 6-mile cleanup, and they removed over 2,000 pounds of trash. And this photo on the bottom here is from one of the dives that they did off of Sand Harbor. So you can see a lot of cans. There's some fiberglass pieces from boats, a lot of different interesting trash that they pulled out of the lake. And by getting that trash out of the lake, while it's larger pieces, it's not going to break down into those microplastics. And if you are outside of the Tahoe area, Keep Truckee Meadows Beautiful is a great organization to get involved with in the Reno area as well. Another thing you can do is support bans, so plastic bag bans or bans on styrofoam. Once again, if we're not using those plastics, they can't get into the environment in the first place. And spread the word, reusable water bottles make great Christmas presents, or bamboo cutlery. So talk to your friends, talk to your family, encourage them to minimize their use of plastics. And that's a good way to make a difference. And I like working on microplastics because I do think it's something that everyone out there can actually, make a difference, because small actions by many of us add up to big change. So bring your reusable bags, use your refillable water bottles, and all that adds up. I want to say thank you to our funders. So our microplastic sink study is being funded by the Environmental Protection Agency with a grant that's managed by the Nevada Division of Environmental Protection. We've also received support from the Tahoe Water Suppliers Association, the Incline Village General Improvement District. And thank you to our 2018-2019 summer interns. They did a lot of the work that kind of laid the foundation for us to do this next big project. Also, Stephen Sesma and Jackie Lang, they're the chemists in the lab running samples up here at Tahoe and down at Davis. And Brant Allen and Brandon Berry are helping out in the field. So this work wouldn't be possible without the support from all those people. Here's a couple citations if you want to look up the work that was done on the Great Lakes or in the Bay Area, and then a couple links to get involved. So that is it from my talk. And I guess we'll open it up for some questions.
Okay, thank you, Katie. Thank you, Jenessa. So I'm going to do my best to filter through the many questions we received on the Q&A board, and thank you for posting those and keep posting them. Okay, so some of them I can answer myself. One question was, how can we keep up with the research? Do we need to wait until results are published? Sadly, yes. As Katie mentioned, preliminary results are interesting, but they can be misleading. But as Katie said, the final report is due in the middle of 2021. And given the interest in this topic, I'm sure we could persuade Jenessa and Katie to come back and give us the answers to these very interesting questions that they've posed. So one question, and this one I guess is for Katie, is, are the sampling and analysis methods used in Lake Erie and San Francisco Bay the same as the ones you're using here?
So the ones for the San Francisco Bay are very similar, very comparable. The one in Lake Erie, they didn't look at fibers, so the numbers are a little different, but the rest of their analysis was pretty similar. So the numbers aren't perfectly comparable, but it's close. And like I said, there just hasn't been a lot of work done in freshwater systems, so I was really having a difficult time finding these numbers to kind of compare Tahoe with other systems. Okay.
And Jenessa, if either of you, if one answers, the other feel free to jump in and amplify if you wish. So a question from Peter: what is the source of microplastics in Tahoe and how can we prevent them? Katie touched on this in her presentation, as did Jenessa. Did either of you want to add anything to that, to your answers already?
I'm happy to add a little bit. So we don't have wastewater coming into the lake. So luckily we can eliminate that as a source of plastic into the lake. So we think a lot of the microplastics coming into the lake enter as big pieces of trash that slowly get broken down over time. There's also atmospheric deposition that can blow like small fibers in from other systems onto the lake. Also stormwater runoff. So tires, as they break down on the road, leave little pieces of rubber, and when you have a storm event, that can wash plastics into the lake as well. So I think those are the primary sources of trash in Tahoe. But feel free to add to that, Jenessa, if there's something that I missed.
The only comment I would say is it's a little early to tell from our data, since we don't have it all yet, to know it, because sometimes we can look at the plastic type and come up with the most common uses for that plastic type. And sometimes that'll give us an idea of maybe where some of the plastic particles might originate. And we don't have that information yet to really draw any conclusions about, what even the population of, microplastic types are throughout the year that, in the different samples that we're getting yet. So hopefully we'll have more information on that next time we present. But right now we don't have enough to really make any, conclusions about that.
So a question here about rubber tires. or synthetic rubber tires, I should say. Jenessa, have you been able to detect fragments, microplastics that are obviously from tire products or byproducts?
Yeah, so what I would say is we haven't necessarily identified one that we are certain is from a tire product. And the use of Raman can be a little bit challenging for tire particles because of their black coloration. But what I will say is oftentimes if we find a particle type that we can't identify specifically with our analytical method, sometimes we can then go back and say, we're seeing a lot of this specific type of particle. Now we need to go find out what that is. And then we can do further analytical techniques to kind of complement what we're doing in order to get the most information out. So if we find a particle that we can't identify and we're suspicious that it could be something like a tire particle, oftentimes we'll do a little bit additional to get that answer.
So, and actually this related to another question somebody had earlier. So when you say the plastic is unknown, does that mean that the libraries that you cross-referencing with just aren't extensive enough, or are there secret products out there that we're not aware of?
So I think it's a little bit of all of that. So sometimes it's a lack of it being in the library. So maybe exactly like a fingerprint bank. If somebody hasn't gotten their fingerprints, put into the database, it's hard to identify them. Same thing with our chemicals. So with that being said, we actually use the libraries that most of industry uses. So the people making the plastics actually use the same library we do. And so most of the plastics out there are represented in the library. That being said, there are tons of new additives and different combinations, especially with dyes that can be incorporated that can sometimes interfere with our ability to get a really representative spectra. And so sometimes with those, we can kind of narrow it down or have a good guess. And then sometimes we'll have to use a different methodology to really get that proper ID. But in general, Raman is going to be one of the best methodologies that's out there. And unfortunately, no methodology that's 100% actually really exists yet for microplastics. So we're doing the best we can to get the information, the valuable and appropriate information out of the samples we get.
Okay, thanks. So one question that I will usurp and answer, and the question was, what effect do these microplastics have on lake clarity? Lake clarity is affected by the very smallest particles. So these are particles in the size range of about 1 to 4 microns. So that slide that Jenessa showed with those very small 1 or 2 micron particles, they are the ones that potentially could affect clarity. But the numbers that Katie is producing so far, the concentrations of them, suggest that plastics are much smaller in concentration than silts and clays washed in and algal particles. So preliminary data suggest they will have a minor role in clarity. Okay, so now there are a couple of questions here that probably Katie might want to have the first go at. And that's sort of, what role do you think things like wind and wave direction and lake currents might have on where microplastics are showing up on beaches? And related to that are sort of questions on, well, actually, why don't we do that one first?
Okay. So I do think that the seasonality of weather probably has a effect of where we see plastics in the lake. So I've been out at Hidden Beach and on the East Shore in the wintertime, and because of our southwest wind direction that time of year, it tends to wash things up on that side of the lake. So I think if we went out and sampled the northeast shore during the wintertime, you may find more plastics because the wind's just driving them that way. Versus on the west shore, that time of year, it might be a little less.
Okay. Just a comment here from Justin Reese, who's a professor at Northeastern University and was tuning in, and he is very excited about using this presentation for a class he is teaching on global oceanic change back at Northeastern. Fleece clothing, blankets, we're all wrapped up in it all winter at Tahoe. Is that ending up in the lake, or fibers from that?
There was some fibers in those that preliminary data that we that I presented. I don't remember what the composition of those fibers were, so I think, you know, pending once the final results from this study come out, we'll have some more conclusive, you know, findings to report. I'm sure they will end up in our samples because we're wrapped up in it when we're out on the boat all winter. But I will say, when we're out collecting the samples, we make sure we're wearing all cotton. So any fleece fibers in our surface samples will not be from the boat crew.
Well, have fun wearing cotton this winter. I guess another question here, are there any plans for snow sampling?
We're not doing any snow sampling, but the Desert Research Institute, I believe the team that's studying microplastics there is currently doing some work looking at microplastics in snow in the area. So check out their work as well. Okay,
so we're at 1 o'clock now, and so we need to wrap this up. Those of you who submitted questions, we have those archived here, and we'll be passing them on to Katie and Jenessa, and I'm sure they will get back to you personally on these. So again, I wanted to thank Jenessa and Katie for their time this afternoon. I want to also thank Heather Segali for getting this organized so well, as she always does. And I wanted to thank you, all you people out there in Zoom land, for tuning in. Again, our next seminar is in 4 weeks, and it's on climate change and what is it on, Heather? Energy and climate change, renewable. Look it up. It's on our website, turk.ucdavis.edu. So once again, thank you presenters, thank you audience, and we look forward to seeing you next time.
Thank you so much.
Thank you.